Hello everyone. So in this video we are going to learn about electron transport chain. The topics that I'm going to include in this video are electron transport chain, introduction of mitochondria, components of mitochondria and the components of electron transport chain. So firstly I'm going to tell you shortly about the synthesis of ATP. ATP can be synthesized in two ways. The first one is oxidative phosphorylation and the second one is substrate level phosphorylation. Generally, phosphorylation means the transfer of phosphate group from one compound to another. So when we see the first one, that is oxidative phosphorylation, it is mainly linked with the electron transport chain. In this, there is a production of ATP from the oxidized NADH and FADH. It is also a major source of ATP in aerobic organism. Whereas in the substrate level phosphorylation, ATP may be directly synthesized during the substrate oxidation in the metabolism. Or you can say that there is a direct phosphorylation of ADP with a phosphate group by using the energy obtained from a coupled reaction. Now we will see about the electron transport chain. The energy-rich compounds like glucose, fatty acid, amino acid undergo a series of reactions, generally metabolic reactions, and get oxidized to carbon dioxide and water. The reducing equivalent from the reaction, generally the metabolic intermediates, are then transferred to coenzyme NAD plus and FAD, which produces NADH plus H plus and FADH2. This two reduced coenzyme NADH plus H plus and FADH2 then passes through the electron transport chain and it reduces oxygen to water. During this process, there is a loss of free energy which is utilized to generate the ATP from ADP and PI. This whole thing takes place inside the mitochondrial membrane. Now, when we see the overview of this ETC, this carbohydrate, fatty acid, amino acid are oxidized to carbon dioxide and water, and during this process, the reducing equivalent which are formed are taken up by this NAD plus and FAD, and then this NAD and FAD are converted to two reduced coenzyme, that is NADH plus H plus and FADH2, and then this two reduced coenzymes enters electron transport chain. During this process, the ATP is formed with the help of ADP and PI, and the half molecule of oxygen is converted to water. So this is the overview of the electron transport chain. So now, before moving forward, we will see about the mitochondria. It is known as the powerhouse of the cell. It is the center for all the metabolic oxidative reactions which produces reduced coenzymes NADH and FADH2, which are then utilized in the electron transport chain for the formation of ATP. That's why it is often referred as powerhouse of the cell. Now, when we see the mitochondrial organization, the mitochondria consist of the five parts, that is outer membrane, inner membrane space, cristae, and matrix. As you see the structure of mitochondria in the video right now, the mitochondria an outer membrane, have an outer membrane that is permeable to most metabolites, and an inner membrane and an inner membrane and an inner membrane that is selectively permeable, and there is a matrix inside them or between them, not between them, within them. And all the components required for the electron transport chain and ATP synthesizing system, all the components are located in the inner mitochondrial membrane. This inner mitochondrial membrane are folded to form a cristae. Because of this structure, the surface area of the inner mitochondrial membrane is greatly increased. The inner surface of the inner mitochondrial membrane possesses the phosphorylating subunits that are the center for the ATP production. Now, when we see the structural organization of ETC, the this inner mitochondrial membrane have five distinct enzyme complexes that is denoted as complex 1, 2, 3, 4, and 5. The complexes 1 to 4 are the carrier of electron, and the complex 5 is responsible for the ATP synthesis. Apart from this complex enzyme complexes, they contain coenzyme Q, cytochrome C, and oxygen. Now, when you see these complexes, it is in the video right now. The complex 1, 2, 3, 4 are the electron carrier, whereas the complex 5 is the complex which is responsible for the ATP synthesis. So the electron transport takes place from complex 1 from complex 1 to complex 3, and from complex 4 it enters this complex 5. I mean to say, when the electron transfer takes place from this pathway, after that, when everything is done, the ATP synthesis takes place from the with the help of complex 5, and this complex 2 directly uh, comes from the other route, and it enters this directly into complex 3. So this was about the complexes of this etc. Generally it is having five types or five complexes. This from complex 1 to 3 4, these are the electron carrier, whereas the complex 5 is the responsible for ATP synthesis. Now when you see the components of the electron transport chain, uh, there are five distinct carriers that participate in the electron transport chain, and that are nicotinamide nucleotides, flavoproteins, iron sulfur proteins, coenzyme Q, and cytochromes. These are arranged sequentially and are responsible for the transfer of electron from a given substrate to ultimately form water after combining with proton and oxygen. So first we will see about the nicotinamide nucleotide. Nicotinamide nucleotide, these are the coenzymes derived from the vitamin niacin, generally that is NAD plus and NAD plus NADP plus, sorry. These are like derived from vitamin niacin, and they are NAD plus and NADP plus. Among these two, NAD plus is more actively involved in the electron transport chain. This NAD plus is reduced to NADH plus H plus by dehydrogenases with the removal of two hydrogen atoms from the substrate AH2. As you can see in the equation, from the AH2, that is substrate, this two hydrogen is removed, and then it is taken up by this NAD plus, so it forms NADH plus H plus. Now the second one is the flavoprotein. The enzyme NADH dehydrogenase is a flavoprotein with the FMN as a prosthetic group. As written in the reaction on the coenzyme, as written in the reaction, the coenzyme FMN accepts two electron and proton from to a proton to form FMNH2. So when you see in the reaction, FMN is ultimately converted to FMNH2. Now the succinate dehydrogenase enzymes found in the inner mitochondrial membrane is a flavoprotein with FAD as a coenzyme. This can accept two hydrogen atoms from succinate and form FADH2 as given the reaction. As given in the reaction, like succinate plus FAD, it forms fumarate, and while the succinate is converted to fumarate, this two hydrogen is converted given to FAD. So the FAD is converted to FADH2. Now we will see about the iron sulfur protein. Iron sulfur protein is denoted as FES. It exists in the oxidized or reduced state. The oxidized state is denoted with Fe3 plus, and the reduced state is denoted with the Fe2 plus. One FES participates in the transfer of electron from FMN to coenzyme Q. The other FES protein associated with cytochrome B to cytochrome C1 participate in the transfer of electron. Now the fourth one is coenzyme Q. Coenzyme Q is also known as ubiquinone. Coenzyme Q is a lipophilic electron carrier. It can accept electron from FMNH2 produced in the ETC by NADH dehydrogenases or FADH2 produced outside ETC. Coenzyme Q is not found in mycobacteria. Now the last one is cytochromes. The cytochromes are the conjugated proteins containing heme group. The iron of heme in cytochromes is alternatively oxidized and reduced, that is to Fe3 plus and Fe2 plus form, which is essential for the transport of electron in the ETC. The electrons are transferred from coenzyme Q to cytochrome in the order of B, C1, C, A, and A3. The property of reversible oxidation reduction of heme iron present in the cytochromes allows them to function as effective carriers of electron in the ETC. So now when you see the electron transport chain, can see on the left hand side there is AH2, which is like oxidized, and it get converted to A form, and during that process the electron transfer from this nicotinamide nucleotide, nicotinamide nucleotide, and then it is again taken to from this nicotinamide nucleotide it is taken to the electron is transferred to flavoprotein, and from that flavoprotein it is taken to cytochromes, whereas this coenzymes and this iron sulfur protein are carriers. These are the carriers of the electron. Clear. And lastly, when it reaches the cytochromes, in the cytochromes also it is in the sequence of as it was already given from B to C1 to C and A3, and finally when it reaches the ends, this oxygen is converted to water and the electron transport chain is completed. So when you see the structure of mitochondria, in an overview of ETC, there is an outer membrane and there is an inner membrane, and in between these two there is an intermembrane space. So the inner membrane contains the components of electron transport chain that are kept in a sequence that, as I've already told you, this NADH is the nicotinamide nucleotide, and then it is FMN is the flavoprotein, and this COQ is a coenzyme Q, and this BCA and A3, these are the uh, what you can say, it is the cytochromes. So in this, iron sulfur protein is not shown because it is a carrier, and in the last section you can see this F1 and F node. It is the complex 5. This is the complex 5 which helps in the ATP synthesis, whereas this 5 components are all together forming this complex 1 to complex 4. So I think by this figure the electron transport chain is clear to you. So I'll just brief about this electron transport chain. uh, Firstly we saw about this sorry wait firstly we saw about the synthesis of ATP. In that we saw there are two types, that is oxidative phosphorylation and the substitutable phosphorylation, and this oxidative phosphorylation is directly related or linked with electron transport chain, whereas substitutable phosphorylation uh, is uh, is not related to electron transportation, and it is a direct phosphorylation process. After that we saw about the electron transport chain. In this we saw about this rich compound like glucose, fatty acid, amino acid undergo reactions of metabolic reaction and get converted to carbon dioxide and water, and during this process this reduced coenzymes are formed, that is NAD plus, H plus and FADH2, which are later utilized for the formation of ATP. After that we saw about the mitochondria and the mitochondrial constituent, that are outer membrane, inner membrane, intermembrane space, cristae and matrix. So in the intermembrane space there are the components of electron transport chain. After that we saw about the components of electron transport. There are 5 components: the nicotinamide nucleotides, flavoproteins, iron sulfur proteins, coenzyme Q and cytochrome. So this was about the electron transport chain, and in the next video we'll be learning about the substitutable phosphorylation and the oxidative phosphorylation. Thank you.